Good morning everyone. So today my seminar will be on the topic the role of oral dysbiosis in periodontitis and systemic inflammation. So to begin with, the mouth is the middle of the body. So as we all know, oral dysbiosis, it is nothing but a microbial imbalance in the oral cavity. So it does not stop at the gum disease itself. So it has a silent systemic link also, which leads to systemic inflammation. So dysbiosis is the central to the onset and progression of periodontitis, which is a chronic inflammatory disease which affects the tooth supporting structures. So, but beyond the gingiva, this oral dysbiosis can serve as a biological bridge to all systemic inflammation, which contributes to many systemic diseases like diabetes, cardiovascular diseases, rheumatoid arthritis, and Alzheimer's disease. So in this seminar, we will be seeing on how there are intricate microbial and immunological cascades that are involved, which links the oral dysbiosis to the systemic pathology. So beginning with the healthy periodontal microbiome, its structure and immunological balance. So there is this microbial homeostasis in health. So the gingival structure, sulcus and the tooth surfaces, they are colonized by gram-positive facultative anaerobes, which include Streptococcus species, Actinomyces, Rosia and Veillonella. So these species, they contribute to the commensal biofilm development, the pH regulation and the cross-feeding of the metabolic groups. So this is diverse yet stable. So this microbial community is supports tissue homeostasis and prevents the pathogen overgrowth. So there is also a immune microbial crosstalk that is involved. So that these have host pattern recognition receptors like TLR2 and TLR4, which detects the microbial components and include the immune tolerance. So the neutrophils, they pack out the gingival margins and sulcus and the sulcus in a non-destructive regulated manner. So the salivary IgA, mucins and defenses, also the lysosomes, they limit the bacterial adhesion and they also maintain balance. So any disruption in this balance by any ecological stressors, it will lead to dysregulated immunity, biofilm maturation and tissue inflammation. So there was a very recent study conducted by Dodson et al. In 2022 that showed that the interleukin 17 producing T cells, they were tightly controlled in a healthy gingiva, but they were overreactive in early periodontitis cases, which indicated a very critical immune checkpoint. So moving on to the dysbiosis and the onset of periodontitis. There is early ecological shift in the biofilm composition. So triggers like plaque accumulation, inflammation or salivary dysfunction, they'll contribute to the emergence of anaerobic, proteolytic and invasive species like Papillomonas gingivalis, Canaverbula forsythia, Treponema denticola and Fusobacterium nucleatum. So all these organisms are mainly said to distribute the biofilm structure so that it enhances the proteolytic digestion of the host tissue and also stimulates the pro-inflammatory cytokines. There is also this key pathogen concept where P. gingivalis, even when it is abundant in very low concentrations, it can modulate host response by producing this gingivates that degrade the complement proteins, inhibiting the neutrophil clearance and also inducting the TH17 skewing and these species, they act as keystone pathogens, destabilizing the entire microbial community. So there is also this immune dysregulation involved. So a prolonged microbial challenge can result in elevated TNF alpha, interleukin 1 beta and interleukin 6 levels. So rankel mediated osteoclastogenesis will also be seen, which will lead to alveolar bone destruction. So this actually sets up a vicious cycle where there is dysbiosis, inflammation, tissue breakdown and there is even more dysbiosis again. So this happens in a cycle. And there was a very recent study in 2023 by Kantasti et al. Which highlighted the role of epigenetically reprogrammed macrophages in sustaining the inflammation in chronic periodontitis. Now, there is this oral systemic axis how the local inflammation different to the systematic, how it is systematically. So like pathogen dissemination which happens first. So daily activities like brushing, chewing or flossing, it can cause transient bacteria. So oral pathogens like Porphyromonas gingivalis, Dysbiosis nucleatum, they have been detected in atherosclerotic plaques, colorectal and pancreatic tumors, and amniotic fluid and placental tissues. There is also an inflammatory mediator spillover where the gingival inflammation, it increases the interleukin, CRP and TNF alpha levels and other systemic markers also. So these cytokines will thereby contribute to insulin resistance, vascular endothelial dysfunction and very low systemic inflammation. So there is also the strained immunity where repeated microbial exposure programs the bone marrow derived monocytes. So these are trained macrophages that will display a high-end inflammatory response in very remote tissues. There was another study in 2022 that provided the evidence that oral dysbiosis, it primes innate immune memory, exacerbating the cardiovascular and the metabolic conditions. Moving on to the disease links, the evidence from the epidemiology and mechanisms. So first we'll be seeing about cardiovascular disease, where oral pathogens, they increase endothelial permeability and they also promote foam cell formation. So this Porphyromonas gingivalis, it increases the VCAM1 and ICAM1 and oxidize LDL uptake by itself, leading to cardiovascular diseases. Secondly, we have diabetes mellitus. It has a bidirectional relationship where periodontitis, it directly exacerbates the glycemic dysregulation itself. Plus this diabetes, it also promotes the AGE accumulation which worsens the inflammation. Third, we have adverse pregnancy outcomes where periodontal infection is supposed to said to have a link with preterm labor, low birth weight and preeclampsia. So Fusobacterium nucleatum, it also triggers the TLR4 mediated inflammation which is said to happen across the placental barrier. There is also a link of this with rheumatoid arthritis where Porphyromonas gingivalis, it expresses a PAD gene causing citrulination of proteins, and that is anti-CCP autoantibodies. Periodontitis patients, they have increased RA activity scores. Next we have Alzheimer's disease where Porphyromonas gingivalis and gingivalis, they were detected in brain tissue and CSF. So it induces beta amyloid plaques, hyperphosphorylation and neuroinflammation in such cases. There was another recent study which confirmed the presence of gingival immunoreactive structures in the AD brains, which reinforces this oral brain axis. So there are some models explaining dysbiosis mediated diseases. So as seen in the previous seminars, we have ecological plague hypothesis where environmental stressors have a major role that promotes the pathogenic consortia over all the microbial communities. Secondly, we have keystone pathogen hypothesis where minority pathogens can disproportionately they disrupt the host microhomeostasis. Thirdly, we have polymicrobial synergy and dysbiosis, also called the PST model hypothesis, where pathogenesis, it arises from all the microbial cooperation. It's not about the dominance. And the Fusobacterium nucleatum, it functions as a bridge species, enhancing the coherent sensing and anaerobic niche formation. And then we have this model called holobiont co-evolution model, where the host and the microbiome, they co-evolve together. So this dysbiosis here represents a failure in co-adaptation, which is worsened by either urban diet or stress or antibiotics. There is another recent study by Ganesan et al. In 2022. They proposed that this Western lifestyle, it decoupled host microbial genetic alignment, which thereby led to immune disorders. Now moving on to the clinical translation and therapeutic prospects. So there is this host modulation therapy using NSAIDs and sub-antimicrobial doxycycline, TNF alpha blockers and interleukin-1 inhibitors. which has this aim to control this hyperinflammation. Secondly, we have microbiome restoration using probiotics and prebiotics. Probiotics like Lactobacillus reuteri, Streptococcus salivarius, they have this potential reducing the pocket app. And prebiotics like arginine supplementation and nitrate-rich diet, they support this NO-mediated microbial balance. Thirdly, we have precision periodontics, where 16S RNA sequencing, shotgun metagenomics, and salivary transcriptomics will offer the risk stratification, and targeted antimicrobial peptide biofilm disruptors and nanocarriers can be used, which are under development. Fourth, we have public health and prevention, where oral health should be integrated into primary care protocols for systemic disorders like diabetes, pregnancy and cardiovascular diseases. And mobile clinics, oral microbiome cleaning, and teleperiodontitis are also emerging solutions. So another recent study showed that combining oral hygiene reinforcement with metagenomic screening, it reduces both the HbA1c levels and the periodontal inflammation and type 2 diabetes. So these are the other supporting studies, where a very recent study by Mulder et al. Showed that the trained immunity is linked to oral dysbiosis. And there was another study by Olsen and Ilmas et al. which stated that Fusobacterium nucleatum it got disseminated to the pregnancy tissues. And there's another study where it was found out that metatranscriptomic shifts were seen in biofilms, in dysbiotic biofilms. So to conclude, healing the mouth may help to heal the body. So oral dysbiosis is not just an oral event, so it's a biological trigger with so many systemic implications. So periodontitis, when understood as a host microbiome co-dysregulation, it offers a model for precision and personalized interventions. So restoring the balance and not just eliminating the microbes, it is the future of oral systemic medicine. Thank you.